Neisseria and Moraxella share some general characteristics. They are diplococci with a kidney bean shape, and they are oxidase positive. Neisseria gonorrhoeae grows on chocolate agar and Thayer-Martin medium, which contains vancomycin, colistin, nystatin, hemoglobin, and isovitalex. It requires 5 to 10% carbon dioxide, and growth may take up to 48 hours. It ferments glucose only. This organism causes gonorrhea, a sexually transmitted infection. It may be asymptomatic and is often mixed with chlamydia. In diagnosis, Gram stain is useful for males but not reliable for females. It can sometimes be confused with Moraxella or Acinetobacter. Cultures should never be refrigerated. Treatment may include penicillin or spectinomycin, but a beta-lactamase test must be performed to confirm penicillin sensitivity. Today, most laboratories rely on probe technology for identification. Neisseria meningitidis grows on blood agar, chocolate agar, and Thayer-Martin medium. Growth is enhanced by 5 to 10% carbon dioxide. It ferments both glucose and maltose. Transmission occurs via respiratory droplets and requires close contact. Clinically, it causes meningitis, especially in children under 3, and can cause meningococcemia, marked by scattered petechiae in the Waterhouse-Friderichsen syndrome. Pathogenic strains are usually of types A, B, C, Y, or W. In bacterial meningitis, cerebrospinal fluid typically shows neutrophils, decreased glucose, and increased protein. Moraxella catarrhalis is associated with respiratory infections. It grows well on chocolate agar and blood agar, but not on McConkey agar. Colonies are hard and may be moved across the agar surface. It is asaccharolytic, meaning it does not ferment sugars. Most strains are beta-lactamase positive and D-nase positive. Let's compare the major groups of streptococci and how they are identified in the lab. Streptococcus pyogenes, or group A strep, is beta-hemolytic. It is sensitive to bacitracin but negative for sodium hippurate, opticin, bile solubility, bile escalin, and growth in 6.5% sodium chloride. Clinically, it causes pharyngitis, wound infections, scarlet fever, impetigo, and post-infectious sequela such as rheumatic fever. Streptococcus agalactiae, or group B strep, shows a narrow zone of beta hemolysis. It is positive for sodium hippurate and for the CAMP test, which is a key diagnostic feature. It is resistant to bacitracin and negative for opticin, bile solubility, bile escalin, and salt tolerance. It causes neonatal septicemia and meningitis, as well as urinary tract infections. Enterococcus species, part of group D, can be alpha, beta, or gamma hemolytic. They are positive for bile escalin and grow in 6.5% sodium chloride. They are resistant to bacitracin and opticin and negative for bile solubility. Enterococcus causes urinary tract infections and endocarditis and is often treated with an aminoglycoside plus penicillin. Non-Enterococcus group D species, such as Streptococcus bovis, are alpha or gamma hemolytic. They are positive for bile escalin but do not grow in 6.5% sodium chloride. They are resistant to bacitracin and opticin and negative for bile solubility. They can cause endocarditis, and Streptococcus bovis is strongly associated with colorectal cancer. Streptococcus pneumoniae is alpha hemolytic, with characteristic water drop crater colonies. It is sensitive to opticin and positive for bile solubility. It is resistant to bacitracin and negative for bile escalin and salt tolerance. It causes pneumonia, meningitis, bacteremia, conjunctivitis, and otitis media. Finally, Streptococcus viridans is alpha or gamma hemolytic. It is resistant to bacitracin and opticin and negative for bile solubility, bile escalin, and salt tolerance. Clinically, it is associated with subacute endocarditis. Neisseria gonorrhoeae is the bacterium that causes gonorrhea, also known as the clap, or simply GC. a classic sexually transmitted infection. This organism requires carbon dioxide and chocolate agar for growth, and colonies may take up to 48 hours to appear. It ferments glucose only, unlike other Neisseria species. On testing, it is oxidase positive, producing a purple color. These key features, clinical disease, growth requirements, sugar utilization, and oxidase positivity, form the essential framework for identifying N-gonorrhoeae in the laboratory. To differentiate Neisseria species, we rely on sugar fermentation patterns. All Neisseria species ferment glucose. Neisseria gonorrhoeae ferments glucose only. Neisseria meningitidis ferments both glucose and maltose. Neisseria lactamica ferments glucose, maltose, and lactose. This simple framework makes it easy to separate these three organisms in the lab.